Thank you. Thank you for the introduction. I'm Rafał, by the way. That is fine. we have very nice robots who do very similar things at our university, so it's very close. Anyway, so thank you. First of all, thanks to the organizers for inviting me, for giving me a chance to present this topic. So this talk, I was actually waiting for it for quite some time because it connects several things that I'm really fascinated about. And as some of you already know, probably from the last cozy meeting, I'm interested in prebiotic chemistry in the origins of life. So in a way, this touches the surface of that. But what we've been working on for a long time is the photochemistry of biomolecules in general. And more recently, I got quite into quantifying the rates, and especially into electron transfer rates. And that's another thing that I find really fascinating. So I'll tell you a little bit how it all connects into one thing. And one of the elements that I was fascinated by and interested in is DNA damage and repair, but from a little bit different point of view that most groups work on. So when you think about DNA, about DNA strands, RNA strands, they are prone to the formation of photodamages with UV, and the most frequent photodamages are these so-called cyclobutane pyrimidine dimers. So TT dimers essentially are formed by the absorption of UV light of adjacent thymines. Usually one of them is sufficient to be excited, and then a cyclobutane ring is formed. That results in the formation of a kink in the backbone. It stalls the activity of the nucleic acids. It makes the recognition very difficult in the cell, so it cannot be effectively recognized by the complementary strand. And what nature does, it actually developed a set of different enzymes, and this is one of them. Enzymes which can repair these photodamages. So this is a photolyase, and what photolyase does, it identifies the kink in the backbone, and then it sucks in the lesion into the active site of the enzyme, and it has a flavin-adenine cofactor. So this absorbs in the visible, and when it absorbs light in the visible, it can efficiently transfer an electron to the TT dimer, and that is sufficient to break initially one of the bonds, and then the second bond of the cyclobutane ring is broken in the hot electronic ground state. So that was explained by many different means. Even in the beginning of this year, we had some very interesting time-resolved crystallography studies about this. But what we've been interested in is a little bit different phenomenon. So actually, this group from Germany, which was led by Professor Wolfgang Zint, who's unfortunately retired right now, but still is really curious about these topics. They found out that if you have very specific sequences, they can actually repair the TT dimers. And the reason is that some of the nucleobases, they act as very good electron donors, and if you have the right order of them, which they anticipated based on the oxidation potential or the redox potential of them, then they can actually sequentially, possibly, transfer the electrons. So that was the hypothesis. And for us it was interesting because we did a lot of work in the synthesis of prebiotic building blocks from the point of view of photochemistry and the selectivity that UV light offers. But we were aware of the problem that once you get the DNA strands and you don't have the enzymes, they will be photodamaged. So this was the idea, how they could actually repair themselves, and possibly there could be some prebiotic selection of these sequences based on the photostability or the self-repairing propensity. And the interesting thing is that when you compare some other sequences like TATT, GTT, they are not so efficient in promoting in promoting self-repair in general. Okay, but the quantum or the general yield, so that's not the quantum yield, but that's actually the self-repair yield in the photostationary state of equilibrium was fairly low. So we hypothesized that if we can actually understand it computationally, maybe we can do some predictions and propose which sequences could do it in a better way. And the reason was also that the experiments were quite expensive at the time. I think we then found a good way to bypass the cost problem, but still we thought, okay, maybe it's worth to do some screening or to do some predictions. So first of all, we wanted to understand this mechanism and see if they were right about it, about these electron transfers from one nucleobase across at least one more or several more. And we started, we prepared a computational protocol. It's a pretty big system for quantum chemical calculations. So we started with MD simulations of the GATT tetranucleotide to select for conformers based on clustering. And that was done by my colleague Petr Stadbauer from the Institute of Biophysics in Brno. And then we did electrostatic embedding QMMM. So we did DFT for the ground state calculations, and we used actually ADC2 for the excited state. So from now on, everything that you'll see in the excited states in these talks is ADC2. We had a little bit of introduction into that by Przemysl yesterday. And the reason why we actually use ADC2 is basically it provides fairly good balance between the LE and CT states. Well, I know it a little bit underestimates the CT energy, but it's quite systematic in terms of ADC2. So we have it under control much more than in the case of time-dependent DFT. And actually, in this protocol in our interface, we can also optimize the excited state minima. We can optimize the geometries of state crossings. So that was what we were after. The big problem with the photochemistry of DNA and the charge separation is that the low energy range of the spectrum is always dominated by locally excited states. So you don't really see the charge transfer states until very high excitation energies, which from our calculations is pretty high. It actually gets close to the photoionization potential of water. Probably we are a little bit biased because we didn't scan so many geometries. We actually did static calculations for this. But still, you can't really populate them directly. And the other reason is that they have very low oscillator strengths. So one of the questions that we are asking was how actually these charge transfer states could have been populated when the experiments used the 290 nanometers of UV radiation wavelength. And the answer is published in this work, and essentially, once you excite the guanine nucleobase, and it has some time to undergo the photorelaxation on its surface, then the charge transfer states go down in energy. So the population of the charge transfer states is indirect. So you don't see the CT state, but you will see it in some of the further examples of other systems, but basically you can then transfer the electron from the excited G, which is a little bit puckered in the excited state, and you end up with a G plus A minus state, and transferring the electron one step further even lowers the energy of the excited state, so of the S1 further, and it doesn't really matter where your hole resides, so where the positive charge is, because what is really crucial is to have this electron on the TT dimer, which is necessary to open up this cyclobutane ring, as you see. So that bond is broken in this conical intersection, and then the other bond is broken in the hot electronic ground state. Okay, so from this point, when we thought we understood the mechanism, we started asking ourselves a question, so can we now use it to predict the photoreactivity? And the first thing was that maybe we can actually find nucleobases, alternative nucleobases, which have lower ionization potentials, which probably would result in lower energies of the CT states, and possibly these CT states could be populated almost directly. And we found one. When you actually look at the left and right, try to find the difference. So it's almost the same, right? So here you have adenine, here you have 2,6-aminopurine, and the difference is here just in this amino group in the 2 position of the purine ring, so here. And that small change, it actually doesn't affect the strand structure so much, because 2,6-aminopurine recognizes uracil thymine selectively, but it changes the spectrum. So the spectrum is redshifted, and that's the optically bright state. but also what you can see an even more pronounced redshift with respect to the charge transfer states. So your CT states now become some of the lowest excited states in those oligonucleotides having 2,6-diaminopurin. So that was very promising, and that was the moment when I contacted my colleagues from the University of Gdańsk and said, guys, this looks good. Do some experiments, please, for this. And they actually did, so you'll see these results in a moment. But what I was doing in parallel to them running the experiments was I was doing the calculations for this trinucleotide, and basically what we saw was that there is a substantial preference for electron transfer in the excited state instead of really going to this minimum of the locally excited state onto 6-diaminopurin. But even if you go here, you can still quite efficiently transfer the electron afterwards because the barrier is fairly low for the electron transfer, whereas the direct photorelaxation actually is associated with a sloped conical intersection, so it takes quite some time for 2,6-D to go to the electronic ground state. And now when you compare it to the biological one, to adenine, adenine preferentially undergoes photorelaxation on its own very efficiently. That's of course the absorber in your trimer in the lower energy range of the spectrum. So majority of the photorelaxation processes go here. And electron transfer is possible to a certain degree, but you don't get so much energetically from it as in the case of 2,6-diaminopurin. So we anticipated it should be very efficient, and that's what we saw with the experiments as well, that it actually is extremely efficient. And the formation of the repaired photoproduct from the damaged trinucleotide is very fast. And what is even more fascinating by this system is that we actually can get up to 92% of repair for DTT, 80-something for TTD, so you have different strand directionalities, but they are quite the same in terms of the efficiency. There is a small difference. And in the case of ATT, you get 80% of photodegradation or formation of the TT dimers. So here we have the HPLC, we have LCMS. We also confirmed it by differential UV absorption experiments, which were all done by Magda from the University of Gdańsk. Okay, so I have some interesting things. So I'll just briefly say that in this paper we also say that 2,6-diaminopurin is prebiotically relevant. So I mentioned this. It was shown to slightly enhance the rate and fidelity of non-enzymatic RNA template copying, and it can be actually formed, its nucleoside can be formed prebiotically, so we have a proof of principle synthesis of this. But staying with the electron transfer story, there is one more thing which I wanted to show to you from this particular type of systems. So more recently we were asking the question if the strand directionality matters, and if the charge transfer and self-repair rates are comparable in these two, and it turns out that not really. That was something that really puzzled us when we looked at the experimental results which were gathered by Corinna, who is now moving to Germany, I think to the Leibniz Institute, if I remember correctly. And she found quite substantially higher quantum yields of DNA self-repair, but also the yields in the photostationary state of equilibrium were much higher for TTAG, so not for GATT. And here the difference is, it looks quite similar, right, doesn't it? But the difference is that here at the top you have the 3' end of the strand, and here at the top you have the 5' end of the strand. And even though they look quite similar, you actually have quite pronounced differences in the stacking pattern. So I'm showing the highest populated conformers here, but when we look at other conformers, we see basically the same. And that stacking pattern, as we believe, actually affects, well, we saw it affects the energy of the CT state, but we also believe it affects the energy of the charge transfer rate. And there are two things about it which I was a little bit worried, which made me worried. And these are the hand-wavy explanations. So we didn't quantify the stacking and we didn't quantify the rate. So I'll show this to you in a moment. Actually, when it comes to the quantification of stacking, you saw the presentation from Mikołaj yesterday, who already has a code that is available, so you can contact us, you can use it. When our website is finally up, we'll also put it there. Now, when it comes to TTAG, so what we see from these results, from the calculations, is that, first of all, the energies, as I mentioned, of the CT states go down, and also this stacking pattern hinders direct photorelaxation of the intermediate states, so the excited G and also the G plus A minus, which are not productive in terms of the self-repair, but the direct photorelaxation is hindered, which means that now the electron transfer process is preferential, and that's why it is better than GATT, whereas in GATT we see that direct photorelaxation is more probable than in the case of this nucleobase. And really the next thing for me to do was to find the best way to evaluate the electron transfer rates from this place. And now I have a question to you. So what do you think is the easiest way to evaluate those electron transfer rates? So Adam is already smiling because he knows the answer. That is a tricky thing. So who of you thought about Marcus theory? So Marcus theory is quite it's quite a popular approach, right? It's used very often to this, but it works very well for equilibrium electron transfer, but not necessarily for photoinduced electron transfer. So you'll see in a moment why. So the way it works is basically you have these two terms. So one of them is the electronic coupling term, and the other one is the density of states weighted Frank-Condon factor. Essentially, that's the vibrational part, that's the electronic part. And our donor state is like this. It's a locally excited state, that's our acceptor state. And you basically think about the barrier, but you don't calculate it directly. You get the barrier height from the harmonic approximation of harmonic potentials. So it is kind of implicit here in this term, right? The problem with this is a harmonic two-state model. It actually assumes constant electronic coupling, so you just take the coupling from this point, from this geometry on the donor state, and it assumes that states A and B have very well-defined minima, so you need to find these minima, and it is really an equilibrium model. So it works very well for equilibrium electron transfer, but not so well for non-equilibrium, and we are working largely in the non-equilibrium regime here. So what we wanted to do was really to validate if it makes any sense to use Marcus theory or any other theory for electron transfer here. And we looked for a good model system. So our model system is a dimer, a stacked dimer of two adenine nucleobases. So one of them is the N7H tautomer, the other one is the biological N9H tautomer. And we selected it because, first of all, it has the two well-defined LE and CT minima in the excited state and the S1 state, and the CT minimum lies slightly lower in energy. So we tested like 40 different base pairs, and none of them actually met this criterion until we found this one. And the reason we wanted to have this was that we wanted, apart from doing the static calculations to evaluate the rate, we also wanted to do surface hopping. So we had very nice introductions to surface hopping, so I won't go too much into the detail here, but we ran 50 trajectories, and you will see one of them in a moment. And these trajectories actually allowed us, maybe not very accurately, but at least, let's say, qualitatively evaluate what would be the expected electron transfer rate in the system. So, intriguingly, 25% of them accessed the charge transfer state, but even though we made sure to actually excite it in the low energy range of the spectrum, so that we populate the S1 primarily or only, not to see some bias from high energy states. And these are really the processes that we wanted to see. So these are examples of two trajectories. We had about 11 or 12. And the electron transfer occurs at about several hundreds of femtoseconds, even starting from 100. But you also get these odd outliers, which contribute to rather to something like maybe half, maybe 1/3 of these trajectories. And this is really ballistic electron transfer, which we can't really model by the rate approach, but it occurs very quickly and very efficiently. So to give you a notion of how it looks like, I'll show you one of the most representative ones. So this one, where we have an electron transfer to the A7H, and now will be the video. So it starts in the n-pi star state of N7H, and in a moment you will see that it will suck in the electron from the bottom base. And it looks like a model example, but actually if you look across the different trajectories that we modeled, okay, so here it is. So that's the hole, which is in red on this nucleobase, and probably it will come back for a moment. So there is a little bit of hopping. But still, it does more or less of what we expected it to do, but many of the other trajectories have a lot of fluctuations and are quite surprising to us. So in a way, we didn't expect such a variety and diversity in them. Okay, can we go back? Thank you. So now, if you evaluate the electron transfer rate through the Marcus approach, you actually get a rate constant which is massively underestimated. So if you want to extract the timescale of that process, that's 2 orders of magnitude too slow, right? So I already told you what are the problems with Marcus theory, so it's quite clear from this point that many of these things contribute. We can't really say which ones are problematic, are the most problematic, but definitely this is not a set of 2 harmonic potentials, is it? Right? So this is our minimum energy path, and we are actually very far from the harmonic behavior. We do calculate the harmonic vibrational frequencies, which you will see in a moment, and they can still be used to predict the rate in a better way. So the next thing which I used was actually Fermi's golden rule. And now, if you worked a bit with rate theory, you'll realize that it's actually almost the same as the Marcus theory. The only difference is that we calculate this term a little bit better. So it's this density, or that's the vibrational part of that. So of course you can do this by evaluating these Frank content factors, but this is a really tedious, time-consuming, computationally intensive task, especially if you have a system like mine. So the way to approximate this is to use the time correlation function, and basically what we need to properly to properly evaluate this time correlation function to get this vibrational part of the rate is we have to express the frequencies and the modes of the acceptor state in the same way as we express the frequencies and the modes of the donor state. So we do the Dushinsky rotation here. And now fasten your seatbelts. So this can be evaluated or approximated through the cumulant expansion, and if you take the first 2 terms, you can actually get a nice analytical expression. So it looks very scary, but surprisingly it's probably the simplest way to calculate and the fastest way to calculate the rate within the Fermi's golden rule. So actually this E, I made a mistake. I should have put here a different letter, but all of these capital letters here are matrices which involve the Dushinsky relation between the vibrational normal modes and frequencies of the donor and acceptor state. And what we get from this is we actually get a pretty decent rate constant. So we get almost 2 orders, well, not 2 orders, but over 1 order of magnitude better evaluation, and we get the timescale of 16 picoseconds, which is already quite close to what we would expect. So it's not perfect, and I'll tell you in a moment why it's or what are the reasons, the possible reasons to it. I still think it's a little bit underestimated, but it's way better. And the final approach, which I was thinking about, is this just RRKM approach, so Rice, Ramsberger, Castle, Marcus. And I thought, okay, maybe we should work in the microcanonical ensemble, because it's a non-equilibrium process, so we should think in terms of microcanonical rate. This is the key problem if you think about non-adiabatic reactions, because you really need to think about the probability of transition, which you can get from the Landau-Zener approach. But I didn't manage to code it before this talk. So the only thing which I did when talking to Jeremy Harvey, who developed non-adiabatic transition states theory in the RRKM regime for spin flip reactions, is we said, okay, probably we have very strong coupling because our transition state is pretty far from the state crossing, and at the state crossing the coupling seems to be high. So the rate which we get from RRKM, with the assumption that we have the probability of transition equal to 1, is like this. So it is a little bit more underestimated. Okay, now so I'm wrapping up. So now the thing is that, as you have seen, the photoinduced electron transfer occurs through several different channels, and that's probably the reason why we underestimate the rate. So we can right now evaluate the rate for one of them. Possibly, if I can still locate one more minimum, we can evaluate it for another one. But all of them, with the ballistic mechanisms, they contribute to the overall electron transfer process. So that's probably why we see it occurring so efficiently through surface hopping. And if you sum all of them, we will probably get pretty close to what we get from surface hopping. But don't do this with Marcus theory, because you can get it wrong by 2 orders of magnitude. So what I find to work properly from these calculations are either Fermi's golden rule or RRKM. So the NumD simulations were done by Christina, and just a very quick announcement, we are actually developing new research directions in my group, including prebiotic activation chemistry and reaction networks, so that's ground state chemistry, and also we work on non-enzymatic RNA self-replication, and we are looking for a postdoc for this particular project for the next year, somewhere in the middle of next year, so let me know if you're interested. If you are still hesitating, we have a very nice group that is getting even more international. And yeah, thank you for your attention.
Thank you very much. We have time for 1 or 2 quick questions.
Yeah, thank you, Rafał, for this very nice talk. Just a short question. Your rates in Marcus theories, or irrespective of the theory, they always depend on these couplings.
Yeah.
And you calculate them with the double zeta basis set.
Mm-hmm.
And they are known to be very basis set dependent.
Mm-hmm.
Do you think is the way to improve the results? Or?
Yes, possibly. So the way those were calculated, that was actually Boyce localization, so I'm thinking about calculating them better with wave function overlaps. I think, well, I haven't done it yet. I think we even talked about it some time ago. This Boyce localization or generalized Mulliken hash is probably not so dependent on the basis set, but it's also a very simplistic approach to calculating the couplings, right? So possibly we can improve the couplings with getting,
But with Boyce localization, you just use the Hartree-Fock orbitals or something like that. Mm-hmm.
So that's a method that was developed by Joe Subotnick, and it's just a properly formulated generalized Mulliken hash. So essentially you express it in terms of electric and transition dipole moments.
Okay, so it's more generalized. Yeah.
Yes.
Okay, thanks.
Yeah, very impressive work. Congratulations.
Thank you.
I wanted to ask, is there, like, from the surface hopping trajectories, is there any structural parameter that is like maybe the nucleobases are kind of being a bit more apart with this charge transfer happening, so that you could also measure how that, like, how structural is changing?
Yes, so very often, I think it actually depends very much on the 2 bases which are stacked, but what we very often see is formation of an interaction between 2 or a little bit more than 2 atoms of the 2 bases in the CT state. So essentially you get some small degree of puckering on each of them, and then these atoms from the rings, they actually approach each other. So you see like formation of a quasi bond if you go to the conical intersection, but it's usually a sloped conical intersection, so it can stay in this kind of contact for quite a long time. Mm-hmm.
Okay, we have to move to the next talk. Let's thank Rafał again.
Thank you.
And then